Just a couple more minutes. We've got quite a few more people coming in. Give it about one more minute and then we'll get started. Nice to see familiar names in the participant list. And I think we'll go ahead and get started. And good evening, and thank you for attending this evening's virtual lecture. My name is Galen Kinsey, and I am the scheduling coordinator at White Mountain Research Center. I will be hosting this meeting as a representative of WMRC and the California Native Plant Society's Bristlecone chapter. This talk will be recorded and posted on our YouTube channel on a later date, and the link to watch will be posted on our website. As a reminder, I ask all participants to remain muted and keep their cameras off during the presentation. We will have a Q&A afterwards where you can either raise your hand with the raise your hand feature or ask a question in the chat. I'm going to let some more people in here. Firstly, I would like to give a land acknowledgement. UC WMRC acknowledges the Numu, Paiute, and Newe Shoshone as the first peoples and traditional land stewards of Paiute, Hunadu, and Koho, the Owens Valley and White Mountains. As a facility managed by a land-grant institution, we pay our respects to the elders and relations of past, present, and emerging. With that being said, I would like to introduce this evening's speaker. Kaleb Goff is a doctoral student at the University of North Carolina. He is a 2022 WMRC Mini Grant recipient, 2022 CNPS Bristlecone chapter Detector Botanical Grant recipient, and a survey coordinator with our partner GLORIA Great Basin. Tonight he will be presenting his work titled Taxonomic and Functional Diversity of Alpine Plant Communities, in which he will discuss his findings that demonstrate climate change's effects on plant diversity and functionality within the xeric alpine ecosystems of the White Mountains. Kaleb, you have the podium.
All right, thanks for having me. Fun to see so many familiar folks in the room here, in the virtual room, and also grateful to hopefully meet some new people. So we'll just jump right in. I'll just try to share and let me know if this goes off the rails. Just one second. How's that, Galen?
That looks great.
Okay, awesome. Okay, thanks everyone. Yeah, thanks for being here once again. My name is Kaleb. I'm in the Chef Plant Evolutionary Ecology Lab at North Carolina State University in Raleigh. So it's a little later here. That's my sort of maybe first disclaimer is I might be a little less sharp than usual, hopefully not, but it's a little later on this side of the continent. I certainly do wish we could all be together in person this evening, but also glad that we can meet in this way. And yeah, just really happy to be here. The title, thanks to Galen for sharing that. So I'll just be able to move right on to it here and just wanted to start just to show you this photo of the top of White Mountain Peak, place we're going to be going back to this evening. So just wanted to start with a little topo map of the talk. Hopefully we won't be getting lost enough to need it, although I see some of my sort of heroes and mentors. I know that we've got some capable hands here amongst us, so can get unlost. I've done my best to sort of prepare this talk for a mixed audience, so I hope this will be sort of more than a seminar. I tried to, include photos of interesting plants and just kind of other interesting photos and questions that I have that I've been thinking about. I guess that's my other disclaimer is that I'm a second year PhD student. And so I'm going to have many more questions than answers tonight. And I'll be interested to hear at the end what all of you think and what your questions are. So just want to start with a little background about myself and about these places that we're going to be traveling to. I want to talk a little bit about the first chapter of my dissertation, because that's really what I'm working on day in and day out right now, doing some kind of bigger synthesis work. And then I want to talk about this chapter that was funded by White Mountain Research Center and by the Bristlecone chapter, thinking about functional traits and functional diversity. Then I will move on to some future chapters and directions. That's something that is also in my mind right now, kind of on one edge of my PhD, looking into the other edge. And I would just say, oops, sorry about that. The goal tonight is just to try to entertain you and get acquainted, introduce myself and introduce this work. So I'll just start with a little background about myself. I went to UC Santa Cruz as an undergraduate, and I spent a bunch of time in the Coast Ranges, walking around and botanizing and becoming a devoted naturalist, and just thinking about why and how there are so many beautiful things out on the landscape, particularly in spring, but really everywhere all the time. And while I was at UC Santa Cruz, I worked on some speciation research, so thinking about evolution and how we get more beautiful things, and just the origination and maintenance of biodiversity. And it took me a whole, much longer. I was an undergrad several years ago. It took me quite a long time to publish that, though it's finally out now. So if you're interested in Clarkia, you can go see that. I got sort of sidetracked into another adventure, and that adventure was sort of wondering how to do the most good for the world, kind of with the skills and talents that I have and some of the opportunities I had at the time. And one answer that I found for that was to be a park ranger naturalist in Yosemite National Park. So working there and just taking people from all over the world on walks and talks and programs, and sort of thinking about this high ridge as it is between art and science. So I think that's part of my background too, just inspiration and science communication and poetry and art. During that time, I spent a bunch of time walking around in the alpine and taking other people up there to look around. So in a lot of places like this, and those sort of scientific questions became a little louder and a little louder in my mind. And I kind of was drawn back, I guess, to try to answer some of those questions in graduate school. I would say that my big questions for my work and for my academic life are all about the origination and maintenance of biodiversity. So again, wondering where all those beautiful things come from, and how their processes are sort of regulated through time and space. I definitely think that mountains are the key to that. And that, thinking about the compressed climatic gradients and sort of compressed climates over short distances in mountains, and the high diversity of mountain places has been something that just gives me quite a lot of inspiration, and kind of the way that we'll be diving into this talk from some of those kind of biggest questions about ecology and evolutionary biology. So I'm going to be saying alpine, alpine, alpine. I know there's a lot of folks there tonight, or there perhaps are a lot of folks here tonight are quite aware of what the alpine is. But just a definition, I'm going to be talking about the alpine as this place, this set of habitats that's between tree lines, so where trees disappear from the landscape, either by latitude or by elevation. We'll be talking more about high elevation habitats, or high elevation ecosystems tonight. These places have kind of common characteristics, or kind of common facts of life, you might say, which is that they have these really short growing seasons. I'm sure we'll have some conversation tonight about sort of this year in the White Mountains, or this coming year in the White Mountains, but short growing seasons, extreme temperatures at time, both in the winter on windswept ridges, but also in the summertime, low nutrient availability, high UV irradiance, so the sun is baking down onto these places, particularly in summertime. I will try to kind of subdivide these areas more and to think about some of what are the kind of microhabitats and kind of subdivisions of these places. But I just want to kind of bring that to your attention, that this thing that we call the alpine is this heterogeneous space, then that it's heterogeneous sort of within one place in the world, but of course across these places in the world. So just a smattering of alpine areas from the Northern Hemisphere, but of course also thinking about alpine areas in the Southern Hemisphere and in tropical and subtropical areas. Maybe you're gazing into these photos right now, thinking about some of the growth forms, some of the plants that are there. So shrubs and cushion plants and graminoids, all of these commonalities. But I think when you look across these photos, you also kind of see some of the differences. And I want to kind of make this argument of thinking about the alpine with this kind of regional bend And our focus today will be on these mountain summits. So that's where we're headed. Zooming in a little bit further, I'll just say this slide is from a beautiful book by Christian Corner that was updated in the last couple of years. So it's worth checking out. It's called Alpine Plant Life. So zooming in to this middle lower pane, to our sort of neck of the woods. This is not me, but a dear friend of mine, but honestly, just for me, it's kind of a question of like, how can someone not be completely enthralled by a plant community that looks like this, right? So if you're looking like into the back edge of the photo, it's kind of like, oh, it's a bunch of rocks and like, maybe there's some grassy bits back there, but then sort of coming down and coming in close. This is sort of belly botany at its best. These alpine areas, particularly in the Sierra Nevada and Great Basin, are dry and windy. They're these xeric alpine areas. They're pretty different from Northern Europe, where a lot of this research has been done, which is sort of this kind of more north temperate climb where things are a bit more temperature limited. They're a bit colder and a bit wetter. Our places really have this sort of xeric touch to them. And I think this kind of thinking about climate change, thinking about these places becoming warmer, sure, but also kind of thinking about precipitation volatility and about extreme weather events. There's a lot of nice climatological literature about expectations that these places are going to increase in terms of how extreme they are. So the frequency and magnitude of extreme events. So these places will get wetter and drier depending on the year, and also warmer. Which leads us into, I think, what is a big question in global change biology and a big question sort of in our time this evening and in my dissertation, which is wondering whether or not alpine plant communities are robust to climate changes. Certainly thinking, from a paleo-ecological background, these places have undergone, repeated glaciations and repeated ice ages, but thinking about kind of going off into the future. And there are sort of two contrasting ideas that both have merit to them. One is this idea that's up on the slides right now, which is sometimes called the escalator to extinction. It's this idea that cold adapted species, whether they be plants or birds or otherwise, are, as climate change gets more intense or as things get warmer, tracking their climatic niches upslope. When they get to the top of those slopes, they have sort of nowhere left to disperse to. And so we see this kind of cascade of local extinctions. Another kind of contrasting idea is this idea that mountain areas are pre-adapted to climatic changes because they include all of these microclimates, and that the vegetation, the climatic niches of vegetation are heterogeneous in these places. And people have gone, and Christian Corner being one of those people, have gone as far as to sort of map out these areas and to say, well, this area is 6 degrees Celsius warmer than the surrounding area. And so there's 6 degrees Celsius of temperature buffering there. And that's as much or it's much more than we're maybe likely to see with warming. So two kind of ideas about whether or not alpine plant communities might be robust to climate changes. And where might I wanted to show these mountain summits here on the left, because we might expect that these processes would be the most apparent on mountain summits. So things are gonna, so to speak, pop off the top. Maybe we go to those tops. Someone thought of that quite a long time ago. Back when I was like in elementary school, and started this incredible global network called the Global Observation Research Initiative in Alpine Environments. We just call it GLORIA, kind of a wacky acronym. But it's this collective or this global effort to monitor plants on mountain summits. It was started in 2001 in Europe, in Central Europe and Austria. And then in 2004, it was brought to North America by Connie Millar, and has since sort of spread all over the world. And we've learned a lot from a lot of these different places. We've learned quite a lot about what's happening with alpine plant communities. I think another thing that's powerful about this network and powerful about this project is just that it takes a long view in these habitats. So these habitats are characterized by long-lived perennial plants. And one of the sort of thrusts of this project is to monitor those places in perpetuity, which inside the confines of scientific funding is sort of hard to figure out what to do, but this is a group of people trying to figure out how to do that. Just zooming into North America, there are several what we call target regions or just study regions inside North America. And there are 8 that we're going to be focusing on this evening right here. I know many of you are coming to this talk from California and Nevada. excuse me. So we've got 8 target regions, and they're shown over here. I'm going to dive into them in detail, but I want to just call into your attention that these are heterogeneous places, right? Like if you're thinking, if you spend any time in the alpine areas of northern New Mexico, or maybe you've even been over to the alpine areas over here in the East Coast, that these places are quite different, and we might expect that their responses to climate change would be different. To date, these places, or this place that we all, or many of us, hold quite near and dear to us, there are no long-term published kind of time series data sets or analyses of alpine plant summit community changes on peaks. That's one of the things that when I was a park ranger, I went looking for, which is just like what's happening with mountain plants here in the Sierra and in the Great Basin and in the White Mountains. And I didn't find a good answer to that question. And so part of this is sort of coming to answer those questions, or at least attempt to. So a bit of the impetus for what I'm doing in the first chapter of my dissertation, which is all about synthesizing changes in taxonomic diversity over a 20-year period. So GLORIA Great Basin is this nonprofit that monitors those 8 study regions I just showed. And there are 29 peaks inside of that area. They've been monitoring these places since 2004. They're in general surveyed at 5-year intervals. So we'll talk a little bit more about the data structure in just a second. I just wanted to quickly pause on this slide to show just kind of the constancy of those environments. So on the left is a picture near Dunderberg from 2004, and then the right is the same plot in 2014 at slightly different angle, but I think you can see, you can find the same rocks and the same plants. So this kind of incredible constancy. One of the things that I think is amazing about this project is that we really resurvey the same exact area every single time. So kind of an interesting thing happening here. So just reorganizing the study regions. Just wanted to reorganize them a little bit here. So on the panel A, you'll see there's the 8 regions, and then there's those little gray dots. Those are the 29 peaks. I wanted to show just kind of like the broad macroclimatic trends over this area over the study period. So B shows temperature and C shows precipitation. So you can see some of that precipitation volatility. And then D shows this proxy for aridity, that's climatic moisture deficit. And you can see even in these macroclimatic signals, so using sort of a coarse-grained approach to the landscape, that this area that we're studying, these 8 target regions have increased in aridity over the study period. So kind of thinking about those changes in climate and then thinking about what the vegetation responses might be. I want to just drive these places into your minds and imaginations slightly better. So I'm going to just zoom in on a couple of them, starting on the best of them all, which is in the White Mountains, of course. Here we are on the silicaceous, metavolcanic, these really interesting metavolcanic and metasedimentary rocks in the high White Mountains, kind of have this gray-brown, some very bummed out or sad field biologists, just kidding, right there. So thinking about this is one of our study regions with 3 peaks in it. And then also in the White Mountains, a study region that includes 3 peaks that is on the metasedimentary limestone or the dolostone in the White Mountains. It has this bright white albedo, this really unique soil chemistry, and of course a unique plant community. So there's the ancient bristlecone pines there in the background of that photo on the left. And then on the right, this sort of emblematic, kind of wildly, the lion in the jungle of our kind of alpine summit world, Townsendia condensata, so a plant that people come up to the White Mountains to see. Zooming across the caldera or traveling across the caldera to the southern Sierra Nevada near Mount Langley is another one of our study regions. These broad, incredible, dramatic, unglaciated plateaus, a lot of this kind of rocky, blocky work to be done in those kind of places. And we're kind of near the southern range margin, or very near the southern range margin of another emblematic alpine species from our area, sky pilot or Polemonium eximium. And then going up to the north, and admittedly skipping over several target regions, up to the rim of the Tahoe Basin. So there's Lake Tahoe in the background of that photo on the left. This is another one of the regions that's been monitored. We were actually there last year, has some amazing, unique species. So just showing 4 out of the 8, but just want to kind of zoom in and show you the just even the heterogeneity between these regions before we get to looking at the data and kind of thinking about some of the results from this chapter. Okay, so let's talk about data. So we have these 8 target regions, or these 8 study regions inside of this study system. There are either 3 or 4 peaks that are within each one. The peaks are arranged along this elevation gradient. If you're a statistician, hopefully you're already groaning. If you're not, then don't worry about it. But there are 3 or 4 peaks that are ranged across this elevation gradient within each target region. One is usually sort of near tree line or sort of near the alpine subalpine ecotone. And then one will also be kind of as close to the highest peak in the range as possible. And then, so 29 peaks in 8 regions, they're subdivided into aspects and contours, which will be a little bit less important for our conversation this evening. So kind of a top view, and then a really kind of the raw material for this first analysis or this first chapter is just a count of the number of species at each time point on each peak. So kind of summing across all aspects and contours. I'm talking to several, I know there's a bunch of botanists in the audience, and so you all know quite well just how challenging and how fun it is to crawl around in you know, a couple hundred square meters and try to find every single possible thing that you can. So this is sort of our raw data. Before I jump into kind of bucking the normal seminar style, and before I dive directly into showing some of the results and some of the analyses I've done so far on this data, I just want to talk a little bit about hypotheses and what some of the predictions might be for what the number of species on peaks would be doing across time. So just backing out again slightly more, or just a little bit further, to think about some of the properties of ecological communities. So there's the number of species, richness. I'm going to keep saying richness, and that just means the number of unique species. There's turnover, so there's sort of a measure of composition or the stability of composition. So if species are added or species disappear between surveys, so that's turnover of species inside of a community. And then there are abundances. And then if you've been in basic ecology classes, there's ways to combine abundance with richness to make these diversity measures. So zooming in on richness, particularly, what might we expect? We might expect species richness increases, and species richness increase might be because it's becoming easier to be an alpine plant on a mountain summit because of climate change. So maybe it's slightly warmer and it's just a little nicer or a little easier to be there. Another reason for species richness increases could be that the time at which it takes species from lower elevations to disperse and establish on mountain summits is shorter than it takes the species that are up there to go extinct. So I just wanted to show some of the lags in this system on the right-hand side. We might expect species richness decreases. So maybe that's because the extinction lag is short, or there's a dispersal lag. Again, then I'll just say these arrows, the flat arrows, are meant to decrease, or thought to decrease the lag, and then the pointed arrows are thought to increase the lag. We might also expect that species richness over time would be stable. So that's a possibility in our world here. That could be for a bunch of different reasons. It might just be that these things are all quite lagged, and 20 years isn't quite enough time to see those changes happen. And we'll get into each one of those. So what have we found so far looking at the number of species on peaks in the GLORIA Great Basin study region? To date, we found that there is no change in species richness over time. So just a little figure showing that. So on the y-axis, on the left-hand side is just a count of the number of species on each peak. On the x-axis is survey year. So these peaks are surveyed every 5 years on average. So some have been surveyed twice, like in Death Valley National Park. Some have been surveyed 3 times, so we just got back to the Tahoe sites. And some have been surveyed 4 times, like in the White Mountains on the silicates. The triangles in this graphic are just the raw data, so just the count of the number of species on a peak in that target region in that year. And then those dotted lines are model predictions. So we have to make a statistical model to encompass this information and try to get results out of it. We don't want to just be sitting here squinting and seeing if we see a relationship between those triangles. So to date, we found that richness is not changing significantly over time. And we've also found, I think, or we've refound that different peaks have different peaks at different elevations have different numbers of species, and different peaks in different target regions have different numbers of species, which I think are two things that you would expect. There's a strong negative relationship between elevation and species richness. So the higher you go, the fewer things there are, the fewer species there are. One question I think that comes from this finding of sort of relative stability over time is whether richness is not changing because every time we go to a peak, we find the same thing over and over and over again. We just find the same plants, the same individuals. Or could there be turnover? Could there be sort of compositional instability behind species richness? So that's where I'm sort of headed with these, or where I've been headed with these analyses next. And got a little graphic to show you. So on the left is turnover. Turnover is calculated by comparing two lists of species. So it's the number of gains and the number of losses between those two lists divided by the total number of species in both of those lists. So it's proportional to the number of species. It makes this really nice metric, and this is sort of commonly used in community ecology, where these are actually like true proportions. So 0.2 would be 20% turnover between two lists, or between two pairs of years. There is no significant or statistically significant effect of elevation, but there is this significant, or time, but there is this statistically significant effect of target region. So different target regions have different amounts of turnover. And I think to answer the question that I posed in the last slide, there is quite a bit of turnover, sort of across all peaks and time points. The sort of intercept, forgive my stat speak, that's sort of what I'm buried in right now, but the intercept for this model is about 11.3% average turnover across all peaks and survey years. So on average, if we go back to a peak, or on average, two lists are about 10% or 11% different. So that bigger conclusion is that there's turnover that's happening behind changes in richness. As always, further questions abound, which is really great. Several of them I'm sort of working on, like exactly, or as we speak, or just before this, I was working on them. So one kind of interesting idea is whether or not these species can be grouped based on shared traits. So can you kind of group all the forbs and group all the trees and group all the shrubs and group all the grasses and sedges? And then can you think about changes in richness or changes in turnover or abundance based on those functional groups? That kind of generalizes our analysis to places that might not have exactly the same species. And then, of course, you know, we've talked about a couple of these parameters of our properties of communities, that is richness and turnover, but abundance is something that I'm really working on right now. And we actually have sort of a finer scaled data set with quantitative abundance that I'm working on for a future chapter. So there's more there. This is a question, or the last bullet point there is a question that has really been thick in my mind, which is whether or not there are certain species that pop out as clear winners or losers. When you do this work, right, when you're a botanist and a naturalist, and you know all these species really well, you really like to just sort of have some names, put some faces to some names, and sort of think about what are the species that are most likely to be lost from a peak, and what are the species that might be most likely to be gained or lost, excuse me. So I'm eager to hear your thoughts about this chapter, and have more to share, but just want to share things that are sort of ready to be shared. We're hoping that a lot more of this can be sort of packaged into a manuscript very soon. I'm working on it every day. Okay, so moving on just a little bit, we were kind of talking about traits here, or characteristics. I want to talk about grouping species based on their functional traits, or traits that have some impact on fitness, or at least that we think have some impacts on fitness. And, you know, it's cool to think about these functional groups, like sure, forbs, grasses, you can just assign those categories to species you already have. But what if you actually went out and measured the traits themselves on species, on the landscape, and use these traits to say something maybe more general about changes in a community over time? This particular chapter was funded by White Mountain Research Center generously, and also by the Bristlecone chapter. So very grateful to be able to do some of this work. And lots of people out there have sort of inspired me to go down this path. So a lot of people working on this. So one cool thing about functional traits is, or about traits in general, these characteristics of plants is that we can sort of put plants on a spectrum. We can sort of put them on this economic, maybe is the wrong word, but that is the word that we use in literature, I guess. But we can sort of put plants, put traits on plants, or sorry, assign plants, we can map their traits onto a spectrum. So in the spectrum is how resource conservative are they? How likely are they to sort of be conserving what they have? So plants that are resource conservative are small. They might have like short pedicels or peduncles. They're kind of holding their parts close to themselves. They might have like tiny leaves, so small thick leaves, and they might not have a lot of leaf nitrogen in those small thick leaves. So those are traits that we can go out and measure on the landscape. Then there's maybe the more resource acquisitive plants, so plants that are taller, plants that have larger, thinner leaves, and plants that have more nitrogen in their leaves, so sort of more nutrients to work with. We can map this plant economic spectrum, or this kind of spectrum from resource conservative to resource conservative to resource acquisitive. We can map this onto plants that we might find in the alpine. So here's just, three options or three ideas of plants that are resource conservative. I think you can sort of just by gestalt see some of their shared characteristics, but there are also things that we can quantitatively measure. And then plants that are more resource acquisitive, that are also sort of in the alpine or very much near the alpine. So we can intuitively map these traits onto the resident species that are there. We can measure them. And using this method, we can ask whether alpine summits are becoming increasingly dominated by a certain set of traits. So kind of to say it a little bit differently, we could ask if the average value across all species in a community is shifting with global change. So are plant communities getting more resource conservative or more resource acquisitive over time? And our hypothesis in this is that they're getting more resource acquisitive. And this also, again, lets us sort of generalize our work. We don't all have, there are different plants different plant species on summits all over the world. And even in this area, there's 350 or so plant species across those 29 summits. So this gives us a way to sort of move away from taxonomic identities and some of the thorny things there, and allows us to think about plants kind of more in terms of their traits. So last summer, I traveled to the White Mountains quite gleefully and gratefully to collect some data. There are these big functional trait databases, but a lot of these unique species that we have in our region aren't in those databases. So we've got to measure those traits in the field. So I wanted to collect data on 6 functional traits that I showed you in the previous slides that map on to resource acquisitive or resource conservative. There are lots of ideas about what traits you could measure, and I think there's lots of good ideas. These ones are actually pretty easy to measure. So we measured vegetative height and generative height in the field using a ruler, nice cheap tool, very usable tool. And then we also clipped leaves. So this is Eriogonum gracilipes, so clipped the leaf from there. We took these from the field, so we'd usually be collecting in the morning. We took these to the field station and we scanned the leaves on a flatbed scanner. Some of these leaves, I'll show you some scans in just a moment, but some of these leaves are pretty hard to smash onto a scanner bed. Then we weighed the leaf wet mass, so trying to do those things sort of at the same time and trying to do those things close to when plants came from the field so that they're still fully hydrated. We tried to use time rather than like rehydrating leaves or doing other things like that. Then they rode in the back of my truck across the country. They ended up at North Carolina State University, and some fantastic undergraduate researchers in the Chef lab. Emma Wilson and Sophie Meng helped me dry these leaves and weigh their dry mass. And so then we can calculate things like specific leaf area, leaf dry matter content, and that kind of gives us the full suite of traits that might tell us something about whether plants are becoming, or whether communities are becoming more resource conservative or resource acquisitive, just for one example. I want to just give a shout out to a field crew. We had such an amazing field crew. So here's us on the left, like pretending not to have as much fun as we were actually having. And then here's us on the summit of White Mountain Peak. These are the things that are sort of priceless. But Malia, Reese was an absolutely incredible undergraduate from UC Davis that was part of the field crew. And then Seema Sheth, my PhD advisor, was also there to help us out a lot. So grateful to have that help on this project. I wanted to just show a few fun photos from the field. So on the left, Astragalus calicosus, and just showing like how we would measure the vegetative and generative heights. So you can sort of see like where the leaves come to, right? We're sort of thinking about how far is this plant extending into the air or into space. And then Phacelia hastata is also one that really, it's easy to collect leaves from. And it's also the kind of difference in height between the generative and vegetative structures, I think you can really see that in that one. Couple of my just favorites. We didn't collect any leaves from Anelsonia eurycarpa on the right. I just really love that plant. And so I just wanted to show it to you, the dagger pod. And then Erigeron vagus is another really high elevation species. Looks like there's like Elymus elymoides in the background, isn't there always? So a couple of other plants with maybe, you could try to think about some of those functional traits. And then some of the kind of highest plants. I could go on this like this forever, but I ought not to. And then here's us on the shoulder of White Mountain Peak measuring Pulsia algida. So just kind of, we went all over the range to do this. I think this is my hot take of the night. I think that someone should study this plant more in depth, and someone should study its physiology more in depth. This is one plant that sort of bucks this idea that plants get more resource conservative as you go higher in elevation, and it also just like has weird sticky leaves, which I think is super cool. A total spitball, but like there's always a bunch of insects that are stuck to this plant's leaves. I don't know what you make of that, but anyways, there was some lab work to be done, and I just really want to give a shout out of gratitude to all the people who like keep the lights on and keep the Wi-Fi going, keep the printer working. This was our kind of flow. So we bring the plants in a lunchbox with wet paper towels sort of as quickly as possible from the field, usually like by 1 or 2 p.m. And then we would get the scanners going and be scanning the leaves. And then we'd have the balances. I got these sort of fairly accurate jeweler's balances to try to weigh the leaf mass. And there's Malia. She was so good at doing these things. Here are a couple examples of the leaf scans. So Woodsia scopulina is there, again, including that ruler. There's Erigeron compositus and Draba. The Drabas were difficult, partly because they're hard to ID, right? We actually have to associate the correct trait measurements with the correct species identities, at least at this stage. And yeah, the other reason it's hard is because it has these just shards of leaves that get totally chewed up and are just difficult to scan. But I think what I can do with these is use a computer program to map the area of these leaves since they're on these nice white backgrounds. So I've got the leaf area measurements that way. So just in sum, we were able to collect and process 676 samples from 71 species. We got to visit 7 of the summits that have been monitored by GLORIA Great Basin, some of them since 2004. We went to most of those peaks twice because there were things that we had difficulty identifying, or there are things that we identified wrong and had to redo, or things like that. And then also, you just can't get them all. What I tried to do is spread the measurements out. So I wanted to get 10 samples from all the species that were present on these summits. And so if there was a species that was widespread that was found on multiple summits, I would try to collect like 3 from down here lower, and then 2 from the middle, and then the rest from the higher. I would try to spread them out across the range. So that oftentimes meant like, oh, we didn't quite get that, so we got to go back. There are some fun next steps, I think, for this project. It would certainly be nice. So I think we could link this now to the changes over time on those summits using a kind of more fine scale abundance data that we have from up there. But I think also we have these very interesting surveys that go down the mountain. And so of course there are more species as you go down those slopes. So that would require getting a bunch more functional trait data, but definitely think that would be an interesting place to go with this project in the future. And then lastly, I'll just say that it was a pleasure to work on this project. with so many people, but just a big shout out to all the folks that work at the Inyo National Forest, Rick McNeil in particular, but also Blake Engelhardt and all the folks down there. That's where the permits for this project came from, and it's been a fun back and forth with those folks joining on the GLORIA surveys and beyond. So definitely grateful for that. Okay, went a little fast, but that's just the way that it is. So a couple of future directions. You better ask me some hard questions after this. There is a bunch more, and I am excited to kind of see where this goes. So I want to just give you a few questions that are rife in my mind. I'm at this point in my dissertation where I need to plan things for the future, and so I'm sort of thinking about this aspect quite a lot. One of them is this question by Walton Green, who works at the Harvard Botanical Museum, about this idea of whether extinction wields an axe or pruning shears. So there's this idea of species with shared evolutionary histories have shared extinction probabilities? So that's something that I have a proposal sort of related to. Also interested in how species can evolve higher elevation range limits. So one idea, right, is that species need to move upslope in response to climate change, or may need to. Are they going to move their upper range limit even higher? Is that going to be an evolutionary process? I think is an important question, and I'm thinking about how Erigeron might be one to help answer this question. I also have been kind of thinking about whether range position is influencing extinction risk, particularly for alpine obligates. So there's some nice examples in the White Mountains and also in the Sierra Nevada. In the Sierra Nevada, I'm thinking of Pedistra, nevadensis, and of Polemonium eximium. They have these they only grow in the alpine in the Central Sierra. And so thinking about, if you're right near the range margin, what are those populations, what's happening with those populations at the range margin? And then lastly, and I'm thinking about this one a lot, especially sort of in the context of this winter, but also in just the context of my short tenure traipsing around in the Sierra Nevada and White Mountains, but thinking about how this extreme climate variation and how extreme weather events might cause community tipping points. So if you buy this result that alpine plant communities might be mostly stable over this period, over the time period that we've looked at least, is that because they're sort of resilient or resistant on a longer basis, like on the basis of several more decades or even centuries, or are we sort of one super extreme event away from some massive change in these communities? Is something that I think could be evaluated experimentally and something that I'm excited to dig into. Okay, so with that, tons of folks to thank already. I've sort of barely started. Special call out to the California Native Plant Society Bristlecone chapter. Special call out, of course, to White Mountain Research Center. It's just a fantastic place to do research, and it's just a great place to be supported. Been supported by some other grants in this time so far as well. And then really excited to show this slide to you. GLORIA Great Basin and all the awesome people who've also been part of this project. Not only the board members and people who have sort of literally held GLORIA Great Basin up. This, again, is a project that's sort of hard to directly fund through the sort of normal academic establishment because it seeks to sort of do the same thing and sort of bear witness again and again and again, which is incredibly powerful. But these people on the screen have sort of gone out of their way. It hasn't been their primary project. They've been doing tons of other things to sort of keep this alive, and I'm super grateful to be part of it. And then lastly, just thinking about the global magnitude and power of mountains and of people going to those places and visiting them. GLORIA Great Basin's supported by volunteer and community scientists across 20 years, and then across this global scale with GLORIA International is something that I sort of constantly thinking of that. So lastly, thanks so much for your thoughts and attention. I'm excited for your questions and for a little bit of discussion. And I'll just say also, dissertation, sure, but also, something that really inspires me is just being able to bear witness to these amazing places and organisms in this crazy experiment, unwanted experiment called climate change for a really super long time to come. So thanks a bunch. Feel free to send me an email. I'd like to hear from you. We are, of course, working up in the mountains this summer, and I'm working up there as the field coordinator for GLORIA Great Basin. So if you're interested in some of those surveys, you can shoot me an email. I have a GLORIA email now. And if you're interested in just wacky ideas or you have questions, feel free to send me an email. So thanks so much.
Thank you, Kaleb. It was nice to see what your research was about. I just coordinate your stays, and it's nice to see a nice visual representation. So what we will do is begin a Q&A session. Please feel free to ask questions through the chat feature. If you can access your raise hand feature, you can use that too. And so we'll try to keep this as organized as possible. If any of the CNPS board members have questions or comments, please ask in the chat, or anyone can directly message me as well. So I'll open the chat here. So Dave Passavoy was asking what the acronym LMDC is.
Yeah, I said it a couple of times, but it's leaf matter dry content. So yeah, hi Dave, and also, yeah, not gonna pull one over with an acronym like that. But it's this ratio of leaf dry mass to leaf wet mass. So it's this kind of idea of investment in leaf density and investment in sort of all the photosynthetic material if you have like fat, thick, juicy leaves. Yeah. I think.
See. I'm sorry, I didn't mean to interrupt you, Kaleb.
That's okay. Yeah.
Let's see, there's a series of questions from Rick McNeil, and they ask, is the plant turnover direction of back and forth, lots of new species of the same species coming and going?
Yeah, I've done the thing that I'm not sure if thanks for the question, Rick. And I've done the thing that maybe you're not supposed to do, or maybe you're supposed to do, which is prepare a couple of slides for the questions that you think are going to be asked. So I'll just show, I have a slide for that. So I wanted to just show two species lists. So compositional stability, also kind of turnover. So here's two species lists from White Mountain Peak, one from 2014, and the next time we got there was in 2021, because 2019 was quite snowy. So there's 9 species. There's no change in species richness if you were to just compare these two species lists. There's 9 species in both lists, I think, unless I counted wrong. Wait, 2, 4, 6. Yeah. But we have Cystopterus fragilis in 2021, but not in 2014. And then we have Tricetum spicatum in 2014, but not in 2021. And the sort of formula that I just hinted at is just to add the number of gains to the number of losses and then divide by the total number of species between these two. So that would get us a 20% turnover, 0.2. And that would sort of place, it on that figure I showed. So yeah, a more direct answer to Rick's question is, yeah, this is species gain and species loss. So species coming into a community and also species leaving a community. It's a nice way to put it.
We have a question from Jim Moorfield, the notorious Jim. Jim, you're welcome to unmute to ask your question if you would like.
Didn't know I was notorious, but thanks. My question was a little easier to speak than to try to type. I noticed in one of your early slides you were showing graphs of plant richness over time, and if I remember right, you said that the change was not significant so far on those mountain peaks. I did notice a slight rise in the curves as time progressed, and that raised the question in my mind, how are you dealing with the question of whether something is actually a new immigration into a site versus just a new detection that was not emergent in previous survey cycles?
Yeah, that's an awesome question. Thanks, Jim, and also thanks for being here. Fun to see you out there, so to speak. So yeah, I think so. There's kind of 2 questions in here. One is sort of, what can we say about the slope of those lines on that figure, which maybe I'll just show it. I'll just show it again. So yeah, I definitely think that there are some sort of detectable positive slopes. This idea of statistical significance is, that there's enough variation basically to make make us sort of uncertain about what the slope of the line is. So I try to show those ribbons are like our confidence intervals. You can imagine a lot of different slopes inside of those confidence intervals. So maybe that's one way to answer your question. I could also, all right, I kind of sort of considered showing just like a graph of the raw data, which is like where I started when I was sort of thinking about all this. And I think it's a nice place to just say that like in a scientific space, like I can't make too much of this. Like I need to use these formal statistical models to sort of keep from lying to myself, I guess, and sort of to keep from sort of spuriously just saying like, I mean, it seems like the slope of the line is mostly positive. So definitely, yeah, an interesting question, and definitely the devil is in the details of that kind of work. Your second question, I actually have prepared slides for, and I think that probably a bunch of botanists that are in the audience that are wondering about this. So sorry, folks. So yeah, there's this idea, or there's this, I think one thing that Jim's talking about is this thing called pseudo turnover. So this difference, if you take two people to the same place and you say find everything, there might be differences between observers in the species lists from the same plot on the same day. So the only reason for that is that the two people are just seeing things differently. Your question, I realize, Jim, is maybe a little bit more about like, can we detect those differences? And I think the direct answer is no. So the best thing we can do is sort of try to manage observer-driven pseudo turnover. And luckily, when I was working through this, and when we were working through this, there was this awesome paper from Switzerland that came out, which I just have right here in case you're interested in it. But what they did was they had all these plots, these 10-meter circular plots from all over Switzerland, and they had all these eminent European botanists from 2014 to 2021 resurvey them all twice, like on the same day, one right after another. And so they kind of had this kind of setup of figuring out how much pseudo turnover there was. And also in this paper, they test what they could do about it. And they find that there's some steps that they can take to reduce the impact of pseudo turnover on making ecological inferences kind of writ large, richness or diversity being one of those. And what they find is that it can be reduced by as much as 17% by aggregating species, by sort of a list of best practices. And so I want to just show, maybe to increase your confidence in this project, but also just to show the work that goes into it. But just sort of the steps that were taken to make sure that this was as close to, as good as we could possibly get it in terms of reducing pseudo turnover and being confident in our species richness results. This is kind of a wild slide, but is it not advancing? Oh, it is. So kind of 5 steps here. One is things that we do when the surveys are happening. One is something that Jan Knocklinger and I and others did. And then a few more things that I did before this analysis. So in the field, we have multiple observers rotating through peaks, plots, aspects. So there's a sort of randomization of observers. But then those are in concert with these professional botanists. And one of the findings from the Switzerland study was that you want to like train competent botanists over time and have those people kind of being the maestros of the whole kind of ceremony. And so in our study, we're having, Jan and many other people, Dylan and others sort of review at the field station each day. Yourself, of course, Jim, you've been recruited to help us and help me for sure in making some determinations. So reviewing those while we're still there, because we don't want to change the data once we leave, of course. And then before I did this analysis, Jan went through every observation and cross-reference every observation we basically ever made, every peak, every year, every species, with notes and vouchers and photos. So kind of going through everything and making sure there aren't mistakes. And then Jan assigned these problem categories. So there was like misidentifications and name changes and additions and omissions, all again based on this evidence that Jan's been stockpiling. And I addressed those through some computer code, so like making changes that are reproducible, so everyone can see exactly the changes that were made and the reasons for them. And then lastly, penultimately, once that was all done, I made these spreadsheets that had one species per tab or per spreadsheet tab, and they had the species list for each year side by side with the species all in the same row. And once again, the undergraduates in the Chef Lab helped me do this a lot to organize this. And then this got sent to Jan and some other folks. And we sort of looked at this and went down the line and said, okay, does this make sense? And then lastly, and I think most importantly, then we did some aggregation. Why is this not coming up? There it is. So we aggregated to the species level. So like removing all the inforanks. And then we removed species that were only seen once on one peak. That's something that's just been done kind of across GLORIA studies that I'm still kind of thinking about. All these are kind of reversible or changeable. And then lastly, we did this sort of like case by case genus level aggregation where it was like, okay, we can't be sure that the POAs on this peak were done correctly. So we're aggregating the POAs to the genus level, for example, in some cases. And I think we did that in a justifiable way. But I'll be eager to hear kind of what you think about it when the paper comes out. So sorry, that was a lot, and I was hoping to get to talk about that. So thanks for setting me up, Jim.
Sure, and yeah, I appreciate your detailed explanation. I was actually getting at something slightly different, which is not stuff that is the result of observer error, but just something that naturally happens when you resurvey one piece of ground over time. Not every species is going to be detectable in every year, even if the observers are 100%, accurate and complete. And over time, I would expect, a few more species to show up that hadn't been detectable in previous survey efforts. And I don't know that you can even control for something like that, because that's, not something that's an observer problem. That's just a natural phenomenon.
Right. Yeah, totally. I definitely think that this idea of like good botanists always find more is at play in these kinds of studies and in this kind of study system. And I think you might be aware of this, or other folks might be aware of this, but many other GLORIA study areas have found like very large increases in species richness over time, sort of contrary to what is emerging from our study regions. And maybe that's a little bit more of a concern in those places where it's like, wow, species richness is just exploding. It's hard for us to sort of temper the positivity of those slopes with, yeah, how much is that good botanist find more kind of effect in play? Yeah, I appreciate that.
And just one last quick note, and I'll go away. Check your slide that you had labeled Artemisia dracunculus. I think you might find it's a different Artemisia.
Okay, thanks, Jim.
Sure.
Brian, I swear I see your hand raised. I will get to you, I promise. Brian, I fielded a question from the chat here because Ian Winwick had asked a while ago. Fantastic talk. I have 2 questions. Are you considering interspecific trait variability? 2, have you considered dispersal traits to help explain or predict species turnover?
Yeah, I love those ideas. My way of getting at intraspecific trait variation. So this is this idea that there are different, that there's variation inside a single trait inside of a single species, right? So we know this as botanists, right? There are just like certain plants that are just way taller than other plants. And maybe that has some kind of biophysical explanation, or maybe it doesn't. Maybe it's just sort of natural variation. My way to account for that, at least like within the White Mountains study region, is to go measure my own traits in the field. So like a lot of people are using trait databases right now. And then 2 was what I sort of maybe kind of glitched at talking about or explaining, which is to try to sample species from multiple peaks if they occur on multiple peaks, to try to like at least get that sort of geographic variation in species traits, at least within the context of like only sampling 10 individuals, which was kind of what was feasible for me in the context of one summer. So yeah, those are great points. The point about dispersal is a great one, and would be so cool to measure some dispersal traits. This season or last season, they were sort of like just logistically hard because I have to teach in North Carolina, like in late August. And so collecting seeds is hard. But maybe there are some interesting other clever ways to measure them in high summer. Yeah, thanks for that.
Mr. Brian Smithers, would you like to ask your question?
Yeah, sure. I'd actually put my hand down because there were so many great questions in the chat.
And I want to get to all of them.
And yeah, why don't you do that? I can pester Kaleb and do on a regular basis. So I think I'll forego my question. Let's get to those chat questions.
Okay. Let's see. Dave has another one about when richness changes, does biomass as a whole change, or is plant biomass conserved just like energy?
That's
based on what he just saw. Yeah.
I don't know about the second part. I guess all matter is conserved, so I guess the answer is yes. But the first answer to your question is quite interesting, and we have some data on just like plant cover. I mean, you know this below from helping us out so much last season at least, or like we know like how much cover, like the cover of vegetation and the cover of rocks and the cover of scree and the cover of bare ground. I've dived into that data a little bit in the context of the summit area section data. So just like the summit data, it's kind of a tough nut to crack. And I'm not super prioritizing it. I guess partly because maybe I'm running under this assumption that I do think that alpine areas have become more green over time. Or have sort of maybe slightly more biomass over time. And there's a really, really cool recent remote sensing paper that just came out, I think from Dov Sachs at Brown University, using Landsat data to sort of prove that point. So yeah, I think and actually focusing on the Sierra and Great Basin, I think I can actually link that pretty easily. But yeah, so I do think that biomass or like green matter in the alpine has increased over time, even at the highest elevations. I'll link that paper for you.
See, we've got one from Eric Engels. This isn't directly related to your study, but are you seeing any evidence of evolution in real time, like increased hairiness or reduced stature?
Hi, Eric. Thanks for being here, and thanks for asking such a great question. Maybe you kind of felt the pull in this talk, but like, I really want to do an evolution chapter or a chapter that's more about eco feedbacks. So I'd love to, chat more about that. My advisor studies rapid evolution in the context of climate change too. And so, yeah, I'm super keen to think about some of those questions. I haven't thought much about them yet. I am thinking about this kind of gradient of upper range limits among closely related species of erigeron. So like, erigeron compositus, I think, or at least in published floras, has like a fixed upper range limit of like 4,100 meters, and Vagus has a fixed upper range limit of about like 4,300 meters. So maybe, it'd be cool to kind of apply that trait perspective, like about leaf hairs or some other cool, something that we really think affects fitness, and think about the kind of relationships between closely related species, but across populations. So I haven't thought about that yet, but that's awesome.
Okay, let's see. We've got lots of questions. They're grilling you tonight.
That's great.
We have one from Corey Cody, and they are asking, what has been observed about the tree line moving up in the target sites? How does tree line movement impact classic alpine plants?
Yeah, that's an awesome question. I know Brian partly studied this as part of his PhD dissertation, and I'll just butcher maybe the results of this kind of huge body of work that I'm not involved in. But tree lines are advancing. They're advancing relatively slowly and advancing in some kind of strange ways. Like, I think one of the findings from the whites is that like limber pines are replacing bristlecone pines at the upper range margin of tree line. And yeah, I would definitely expect that, I think over centuries, more than over like decades, if there are trees in the alpine, that trees sort of have a big effect on the other plants that are there. One interesting thing about this, or about that, I guess for me, are the kind of whole biotic interaction lens. interactions between plants in the alpine is whether or not interactions between plants become increasingly facilitative or facilitative as you go higher in elevations. There's this idea called the stress gradient hypothesis that as you move upward in elevation, the strength of competitive interactions decreases and the strength of facilitative interactions increases as you go higher up, which is why I sort of stopped myself from saying like, oh man, trees would like totally mess up the alpine or would like totally mess up alpine plants, maybe like at least like tiny little trees as they are when they sort of begin their life, would actually maybe like facilitate plants that are around them in those harsh environments. So total spitball answer to your question, but maybe that's just a little bit of extra context.
Oh, Dave has another question, and I'm not sure if you answered this previously about when there's change in species occurrence over time, does biomass as whole change, or is plant biomass? So we've covered that.
I see that twice, yeah.
Okay. So we have a question from Brett Lovelace. Yeah, I'll let you wet the whistle. So Brett asks, forgive me if I missed it, but does your sampling protocol account for access bias? Just curious how much of your results are influenced by proximate anthropogenic influence, e.g. proximity to trails, etc. And then the second one, are you attempting to analyze any correlations between richest trends and climatic trends? And he's also has a little asterisk blurb on the end if you'd like to read.
Hi, Brett. Thanks for being here. Hopefully you're cozy in the snow. So the protocol is sort of like from this international level, like the kind of one of the big ideas behind the GLORIA international network is to have the same protocol be used in a bunch of different places so that there's a sort of international comparability. Big international comparisons haven't happened yet, but I think they are possible as data accrues. They account for access bias or from those proximate anthropogenic influences. So we purposefully try to put or establish GLORIA sites on places where people are less likely to go. So yeah, like if there's like on White Mountain, I kept saying White Mountain Peak and like I showed a picture and it looked horrible. That's not where the site is. It's not in that place where people do their cartwheels and take Instagram photos. It's like kind of down and away on the shoulder in a place where only gray crowned rosy finches go to. So I think it's protected. In that case, it's protected from that. Sort of another, on the other end of the spectrum, we were just in Freel Peak on the edge of the Tahoe Basin, and that peak, like the actual summit area, has a little bit more. But I don't think there's like a systematic access bias across the sites. There's some, it's kind of haphazard, but with a push towards to try to keep them away from places where people would want to go. So like it's never on like the highest, raddest, craggiest peak in the range. It'll be like kind of on a nub that's off to the side. And then I've done kind of a less than amazing job of putting the richness trends with climatic trends. So like we have these temperature loggers that are buried on the peaks, but they're spotty. Like the data doesn't all line up perfectly. And so just like for the sake of this PhD chapter and like for the sake of this manuscript, I don't think I'm going to try to do a super in-depth connection of like putting all that logger data together and then correlating that with the richness trends. Of course, it would be so cool. It would be so cool to be able to say that there has been increases in temperature on those peaks, and sort of in light of those climatic changes, we still see this stability over time. Like stability in the face of climate change, I think would be a really cool message, but that we don't have the data. We have the data, but it's not prepared enough to do that kind of analysis in a way that I think would be really useful and cool. So I'm just using this macroclimate data, but it's out there. And if anyone is like really good at doing that kind of thing, like imputing climate data from loggers and like trying to play the timeline mishmash game, we'd love to hear from you. We don't really have anyone working on that right now. And then, the temporal reference data it's a great point too. And I mean, there are other ways I could be more clever, I think, about going for the climate data. And I actually have someone on my committee who is a climate scientist at USGS here in North Carolina. And so I at least like have a climate person that hopefully at the next committee meeting will be like, you got to do this. So yeah, thanks for that question. The next question I see is just, how do grazers affect these plots? This is a great question, especially if you've seen alpine areas in other places on Earth, like if you've been to Europe, where like alpine areas in Europe are just incredibly impacted by grazers in a lot of cases. Here, I think I'm not going to say this, hopefully, to be too confident in saying this, but I don't think that the grazing impacts are super intense. The biggest in our sort of amongst our 8 target regions, the biggest presence is probably from marmots and also from the Sierra Nevada bighorn sheep in the White Mountains, the desert bighorn sheep, which we have seen up there. And I've seen grazing, we note grazing impacts on our data sheets. There are definitely grazing impacts that are there, but that they're not like from big domestic ungulate herds in the current day. But I should say that there's this sort of idea in community ecology in the Sierra Nevada, particularly, that like in the late 1800s, during John Muir's time, when there was herds of 10 or 20,000 domestic sheep in Sierra Nevada, especially in subalpine meadows, that did cause an alternate stable state, that there was like a threshold change in the status of those communities. I don't think that's true like right on mountain summits, like within the top 10 meters, but lower down, I do think that sheep grazing in the 1800s has an effect on what we see in the landscape in 2022.
Okay, well, forewarn everybody, I scheduled this meeting to end at 7:30, so it's just a time check with questions. I do have some announcements to make at the end, and as Kaleb said, he's welcome to receive inquiries as well regarding this evening's presentation, or you can email them to me and I will pass them along to him. I think Gabriel Goff, geez, are you related to that person?
Yeah.
I have a question.
I seated the audience with great people, I guess.
And they ask, what kind of modeling have you done with your data to investigate potential future scenarios or events the alpine may or may not experience?
Yeah, I guess I'm doing some kind of like mixed modeling approaches, like at least for the statistical analyses. So like trying to, take fixed and random effects into account and try to be as good about that as possible, and go to a lot of sort of statistical consultings and whatnot. In terms of like making predictions, a big thing to do in this field is to, try to like do a species distribution model. So like take where species are and then project that into the future, or take the climate space that plants are in right now, and predict how much climatic suitability will be in the future. And all those papers predict like 99% turnover in alpine plant communities, like absolutely totally different places. There's one cool example from the Olympics where they do this with 5 Olympic alpine endemics, and they basically have the climatically suitable habitat in future climate scenarios is basically nothing. And this has been done in Europe. It's actually, I think, also was done in the White Mountains in like 2007 or 2009. Van de Ven, I think, is the citation for that. So that's been done. And I think that just the cool question is like, well, how come we don't see that, right? Like, or how come we don't see kind of like the beginnings of that? And I think that's something that, the only way to really, I like the idea, it's this kind of emerging idea in ecology that like we should make predictions for shorter predictions or less long lasting predictions, so that we can like go out and see if our predictions are right or wrong, and then think about why they were right or wrong. So I think I'm more in that camp. And I think this work that we're doing, or this dissertation work, but also just the GLORIA work is one to sort of say like, okay, well, let's go to the field and see if those predictions from species distribution models hold. Yeah, so that's a cool question and a cool avenue.
There are also some accolades in the chat for your work. So I would like to say that this is the most questions we've had in a Q&A. So thank you, everybody. Sometimes it's a little disappointing, but thank you so much, everyone, for your participation. It's really appreciated. So accolades from Jim. Rick McNeil is offering his help if needed. Accolades from Brett. I will let you know that we have another talk next Tuesday. We're just cramming everything into like a 4-week period. And that's the 28th of February at 6 p.m. via Zoom. And that's with local independent rock art researcher and former WMRC staff, David Lee. And he'll be discussing his research on Eastern Sierra Indigenous rock art. And that's free and open to the public. You can register at our website. Our full lecture schedule is also posted on the website. We do also have another botanically themed lecture next month with another WMRC Mini Grant recipient. That's Perry Lee Pipkin with Claremont University and Cal Botanic Garden, where she is completing a floral inventory of the Silver Peak Range in Nevada. And we are also still accepting applications for this year's Mini Grant awards. If there are any students interested in doing research at our facilities. So, and this talk is being recorded. It will be posted. I will likely send out an email to attendees if they would like to watch this again. So let me see if we have any more questions. Everyone is thanking you. Yeah, so yeah, feel free. We have a little bit more time for questions. If not, I'm sure Kaleb is probably getting tired. He is on the East Coast. And thank you very much. You've been a pleasure to work with. You're very good about being in communication with us, and your contributions when asked are really appreciated. So last call for questions. Brian? So, all right, he's like, no, just cut it. Okay, well, with that being said, I will go ahead and wrap up for this evening, and you are all welcome to send inquiries to the email address that I put in the chat, or if you are in contact with Kaleb. So, yeah. Oh, hi, Brian.
Thanks, everyone.
All right. Go, and I'm going to end it. So I will see you all later. Thank you so much.
Galen.
Thank you again, Kaleb.
Yeah, thanks for having me. My
Really
pleasure.
good talk. Thank you. Okay. Good night, everyone.